Hello everyone, I'm Kyowon Jeong, a postdoc in Kolbach Lab in University of Tübingen in Germany. Today I'm going to present FLASHIda, intelligent data acquisition for top-down proteomics that doubles proteoform ID count. Before I begin, I would like to thank to the organizers for giving me such a wonderful chance to present my work in this ISMB conference. Let me quickly review top-down proteomics as compared with bottom-up proteomics. This sausage link illustrates an intact protein molecule and each sausage a peptide. In conventional bottom-up proteomics, the protein is digested into short peptides, and from each peptide, MS2 spectrum is generated. On the other hand, in top-down proteomics, intact protein is analyzed as is without digestion. Top-down proteomics is therefore suitable to study proteoforms. Proteoforms are the different forms of a protein produced from a gene with genetic variations or post or co-translational modifications. Since proteoforms provide higher resolution and phenotype closer information than proteins, they are gaining more attention in medical and clinical researches. And because top-down proteomics provides so-called bird's eye view of proteoforms, as compared to bottom-up providing relatively narrow sight of proteoforms, top-down is now becoming the method of choice for proteoform resolution studies. But top-down proteomics still has many challenges to overcome in all different modules like sample preparation, instrumentation, and data analysis. Out of all difficulties for data analysis perspective, the complexity of the signal is the most significant source of the problems. Given a proteoform with mass m, its signal in mass dimension is already quite complex due to its long isotopic pattern. And when this molecule is ionized, we observe multiple isotopic patterns with distinct charges in the mth dimension. So given the raw signal without annotation, reverse engineering to the original mass is a very important task in top-down proteomics analysis, which is called mass deconvolution. The complex signal structure also complicates the data acquisition in complex samples. For instance, look at this annotated MS1 spectrum containing two proteoforms. The red and blue ones are the annotated peaks from the two proteoforms respectively. For this signal, if an instrument uses top 4 intensity acquisition, they are the ones that are selected. Even if the blue peaks form perfect charge distribution and isotope patterns, none of them will be selected in this scenario. If we have deconvoluted masses from the previous complex raw signal, the acquisition becomes far simpler and distinct masses can be easily selected. Another issue is the quality of precursor envelope. This illustrates a perfect isotope pattern of a proteoform of a specific charge. We always want to select such envelope for MS2 generation. The bottom range indicates the isolation window covering the whole signal. But even if the envelope is so poorly shaped and with very suspicious quality, if it has an intense peak, it would be selected anyway using conventional data acquisition. And even worse, what if the target envelope is perfect, but within isolation windows, another envelope is present? This may lead to correlated MS2 and possible false positives in proteoform identification. One good idea to address above issue should be to perform deconvolution very quickly during data generation and perform MS2 acquisition with deconvoluted masses. Several methods have been introduced based on this idea, but still long processing time of deconvolution is a problem. Our group recently presented FLASHIda that enables ultra-fast spectrum and feature deconvolution. The runtime of FLASHIda is only a few milliseconds per spectrum, even in complex LC-MS datasets. So with FLASHIda, for this usual duty cycle, which has 1 or 2 second duration, the convolution can be plugged in without much penalty in data acquisition. Or even better, it can be done in parallel, controlling the acquisition in the next cycle, removing any possible waste in the acquisition. After we have the masses real time, thanks to FLASHIda, we still need to decide which masses to trigger. The goal for complex data analysis is to maximize the number of unique identified proteoforms. If we apply the methods used in bottom-up data acquisition, we may adopt top n intensity selection with an exclusion list. In our case, it is actually a mass exclusion list. But still, we have a few issues. First, one may wonder if the intensity is still a good quality measure given the complex signal structure of proteoforms. And second, this simple method still does not guarantee that co-eluted precursors are avoided. And lastly, based on our observation, the simple mass exclusion list was turned out to be too harsh. It diversifies the selected masses, but at the same time, it dropped the ID rate of the MS2 spectrum from them. We present FLASHIda, a data acquisition method that addresses all the above issues. The upper figure shows the FLASHIda AD duty cycle. And this figure shows how FLASHIda works. First, ThermoEye API provides the MS1 full scan to FlashDeconv. FlashDeconv deconvolutes the spectrum real-time, transforming the mg intensity spectrum into mass intensity spectrum. Then the Q-score calculator that measures the quality of precursor isotope envelopes, the spectrum is finally converted into mass quality or mass Q-score spectrum. On the transformed spectrum, top n Q-scoring selection instead of intensity selection is performed, with a quality-based exclusion list instead of simple mass exclusion list. The selected precursor envelopes are then sent to the machine again using ThermoEye API. Q-score is the probability that the mass is identified, estimated by a logistic regression. To measure quality of precursor envelopes, we go back to the original signal. For instance, here the signal from each mass is color-coded. From each signal, relevant 6 features are first extracted. The description of features can be found in our FLASHIda poster in session B. Then with the logistic regression, the feature weights are learned, and with the learned weights, Q-score is calculated. One of the features, we use precursor signal to noise ratio, or SNR, the SNR within the target precursor envelope MZ range. The envelope range is specified by red arrows in this figure. From the observed signal and noise components are separated to measure the SNR. This feature is very useful to avoid co-eluted precursors because the co-eluted precursor signals are considered as noise components. Next, let me explain the quality-based exclusion list briefly. FLASHIda internally stores the masses and their Q-scores over a short retention time period. Then using them, it calculates total Q-score or TQ-score for each mass. TQ-score is given by this formula and means the probability that at least one of MS2 spectra from a mass is identified. For simplicity, we assume the spectra are identified independently. After calculating TQ score for all masses, a threshold is applied. And the ones exceeding threshold are excluded from acquisition for a short time period. Now let's see the results. For the test, 4 sets of E. coli lysate single runs are used. They were generated by thermal eclipse machine. Out of 4, 2 are by FLASHIda acquisition and 2 by standard top-down acquisition provided in the machine. For each acquisition, one set has 30 minutes and the other set has 90-minute retention time gradients. And each set consists of technical triplicates. From each dataset, proteoforms were identified by topic coupled with flash deconvolution at 1% FDR. This bar graph compares the number of identified unique proteoforms with error bars. Blue and light blue bars are from FLASHIda, and red and orange from standard. If we compare 90-minute runs, almost twice more proteoforms were identified by FLASHIda than by standard. If we compare FLASHIda 30-minute and standard 90-minute runs, the numbers are comparable. I also want to remark that this number 800 from standard 90 minutes OLED is higher than most of those reported in literature from equalized singular runs. In terms of proteins, again, significant boost was observed for FLASHIda runs. Overall, the results show that FLASHIda almost doubles proteoform count or significantly boosts machine throughput. It also yielded 30% more proteins. Next, we compare the mass range from FLASHIda and standard. The upper blue bars are the FLASHIda results and red the standard. The light portions are the identified spectra and the dark portions are identified unique proteoforms. It is shown that FLASHIda resulted in more identifications in high mass ranges, but still the largest masses for both methods do not exceed about 40 kilodalton, showing top-down proteomics still has issues in large proteoform identification. We also compare the dynamic ranges. For this figure, the blue bars are the identified spectra and red bars unidentified spectra. If we focus on the blue bars, we can say that FLASHIda in top panel has almost 5 orders of magnitude in dynamic range, between 10 to the 5 to the 10 to the 9. Standard in bottom panel has about 3 orders of magnitude, between 10 to the 6 to 10 to the 9. Lastly, if we compare the areas of blue and red bars, we can see that identification rate is much higher in FLASHIda than in standard. Again, confirming FLASHIda selects high quality precursors. In summary, FLASHIda enables the selection of high quality precursors from diverse proteoforms, and it significantly boosts performance without a major change in experimental setup. The change needed would be to request thermal IAPI license from Thermo and update the machine software. FLASHIda increases mass and dynamic ranges. With this, I'd like to thank to my PI, Oliver Kohlbacher, and colleague Ji-hyung in our lab in University of Tübingen. This work was done with Ole Jensen lab in University of Southern Denmark. I really thank to Masha and Vladimir and Ole. We really created great synergy together. I also appreciate all the funding resources. FlashDecomp is a part of OpenMS software, and FLASHIda is freely available from GitHub site. Thanks a lot for your attention, and I will gladly take any questions. Thank you.